Students, today we'll start with a known membrane bound organelle, that is ribosome. So, ribosome is non membrane bound organelle and it is granular structure. Ribosome is also known as engine of the cell because it is mainly involved in protein synthesis. It is mainly involved in protein synthesis. Now, it is granular structure, non-membrane bound structure, observed under electron microscope since they are very small in size. So, they are observed under electron microscope as dense particles. These were first observed by George Pallade, hence they are also known as Pallade's particle, right? It is non-membrane bound organelle. Now, what are the components of ribosome? Component of ribosomes are RNA and protein. RNA means ribonucleic acid and protein. Ribosomes are present in all the living cells, that is prokaryotes or eukaryote. Exception is mammalian RBC. In mammalian RBC nucleus, all the membrane bound organelle or ribosomes are absent. So it is present in all living cells except mammalian RBC. Ribosomes are the smallest cell organelle since it is present in mitochondria and chloroplast. So it is also known as organelle within organelle because it is present in mitochondria and chloroplast. Now, prokaryotic and eukaryotic ribosome. Prokaryotic ribosome and eukaryotic ribosome. Prokaryotic ribosome is 70S type. Eukaryotic ribosome is 80S type. Now, what is S? S is Swedberg's unit or sedimentation coefficient. It was discovered by Swedberg. So, on the name of the scientist, it is also known as Swedberg's unit, or it is known as sedimentation coefficient, on which unit it is getting sedimented. So, basically, it is indirectly a measure of density and size. So that is about S, the unit of ribosome, which is Swedberg unit or sedimentation coefficient, right? So we talk about 80S type of ribosome. Ribosome has two subunits. Suppose this is the larger subunit and this one is smaller subunit. This is larger subunit and this one is smaller subunit. So in 70S type of ribosome, larger subunit is 50S and smaller subunit is 30S. Together they form 70S ribosome, and association of larger and smaller subunit requires magnesium ion. And at what time these subunits are associated? Association take place at the time of protein synthesis. At the time of protein synthesis. If they are not involved in protein synthesis, then they are independent units, right? So, eukaryotic ribosome, which is 80S, it has larger subunit as 60S and smaller subunit as 40S. Now, ribosome, since we have talked about, it is made up of RNA, which is ribonucleic acid, and proteins, right? So 80 different type of proteins are involved for ribosome formation. 80 different type of proteins. Now this is very important. This question directly came in NEET 2023, right? How many different type of proteins are used for ribosomes? So 80 different type of protein. rRNA, type of RNA which is involved in structural form of ribosome, that is rRNA. Now what is rRNA? rRNA is ribosomal RNA. This ribosomal RNA or rRNA is structural component of ribosome, right? Last one important point, that is about components. If we talk about larger subunit and smaller subunit, then in prokaryote 50S type of larger subunit is made up of rRNA type, 23S rRNA and 5S rRNA. These are the units of rRNA, right? 30S rRNA, 30S subunit is formed of rRNA 16S, right? That is for prokaryotes. For eukaryotes we have 60S and 40S. 60S type of subunit is composed of rRNA 28S, 5.8S and 5S, and 40S is composed of 18S. So these are type of rRNA which is involved in structural component of ribosome, right? So that is all about structural form of ribosome. And now we'll discuss about functions of ribosome. That is one important function of ribosome which is very very important. Ribosomes are involved in protein synthesis, right? Hence also known as protein factory of the cell. Hence also known as protein factory of the cell, right? So that is all about ribosome, which is non-membrane bound organelle. Next non-membrane bound organelle is cytoskeleton. Now what is cytoskeleton? It is network of filamentous proteinaceous structure. Now where cytoskeletal elements are present in cytoplasm. What is the function of cytoskeletal elements? Function is it provide mechanical support to the cell, next it provide motility to the cell, and it is also involved in maintenance of the shape of the cell. So these are the function of cytoskeletal element. Now we'll discuss about how these functions are performed and which component is responsible for these functions, right? So first component of cytoskeletal element is microtubule. Second is microfilament, and third is intermediate filament. So microtubule composed of tubulin protein. Now, where tubulin protein are present, tubulin proteins are mainly involved in formation of spindle fiber, right? So this helps in cell division. How it helps in cell division? By forming spindle fibers, right? So that is a function of microtubule. Next is microfilament. Microfilament are composed of contractile protein. These contractile proteins are actin-like proteins, which is involved in muscle contraction. Since they are useful in contraction and relaxation, so we can say microfilament helps in cell movement, right? Cell movement, or we can say motility by contraction and relaxation. Next component is intermediate filament. Intermediate filament are they form basket like structure around the nucleus. Basically, intermediate filament provide shape to the cell. So it helps in cell division, right? It helps in motility and it provides shape to the cell. So these are about cytoskeletal elements which is present in the cell. Next is next component is microbodies. Now, microbodies are membrane bound organelle. It is single membrane bound structure, vesicles, which is present in both plant and animal cell, right? So we'll discuss about type of microbodies which is present in plant and animal cell. These microbody which are present in plant and animal cell, they contain various enzymes for metabolic activity, right? So type of microbody. Number one, it is peroxisome or uricosome. Number two, glyoxysome. And number three, spherosome. Now, what is the function of peroxisome, glyoxysome, and spherosome? Peroxisome is the organelle which is involved in photorespiration. In photorespiration, three organelles are involved. One is chloroplast, second is peroxisome, and third is mitochondria. So, peroxisome is the central organelle which is involved in photorespiration, or this photorespiration is also termed as glycolate pathway. Now, what is photorespiration and in which plant it takes place? Photorespiration takes place in C3 plants, right? When light intensity is high or O2 concentration is high in plant cells, so plant will escape carbon dioxide fixation and it undergoes mechanism or a pathway, that is glycolate pathway, which takes place only in C3 plant, right? It is also involved in beta oxidation of fatty acid. It is not very important for NEET, but this you have to remember that it is involved in photorespiration, because this point or this topic you will learn in detail in photosynthesis, right? Next is glyoxysome. In plants, glyoxysome is present in fatty seeds such as castor seeds. Now, what is the function of glyoxysome? Glyoxysome is basically highly specialized peroxisome which helps in conversion of fatty acids to glucose at the time of seed germination. At the time of seed germination, glucose is required and this glucose is converted by fatty acid, and which organelle is involved in that? Glyoxysome, right? Next is spherosome. Spherosome is involved in synthesis and storage of lipids in plants. So these are all about microbodies. Now, which components are left in this chapter? One is structure of cilia, flagella, and centrosome, which we'll be doing in next lecture. So first we'll discuss in next lecture, that is cilia and flagella, and next component will be centrosome. So these component we'll discuss in next lecture, and along with that we'll also discuss structure of nucleus.